we're going to talk about Sonic Hedgehog, and this is by far my favorite one. I almost want to get like a tattoo of this Sonic Hedgehog gene monster. Uh, so, obviously, it says nothing to do with what Sonic Hedgehog is, but it's pretty dang sweet. All right, let's do an example of what Sonic Hedgehog is. So in this case, so there's slight differences here between what we see in Drosophila or insects, Drosophila being like the main case, and vertebrates. So I'm gonna kind of focus a little bit more on the vertebrate side here, just because I am sure you're a little more interested in that. Really, the gene names are a little bit different, but the actual mechanisms are pretty much the same. So, if I remember right, IHOG stands for interference hog, and then BOI for Drosophila stands for brother of interference hog. So these are like two dimer proteins. So in the case of the vertebrates, those are named differently. So we have GAS1 and the Bach CDO complex. So these are these membrane bound proteins. You notice they're not transmembrane proteins; they're just membrane bound proteins, and those help to catch Sonic Hedgehog. However, we're not at this point. We're going to look at what happens when it's not active. So nothing's bound here. So what happens is because nothing's bound here, patched, which is this transmembrane protein here, is able to repress smoothened. In the case of Drosophila, it's degraded, and I'm not sure if it is in vertebrates or not. It doesn't matter. It's just not active, right? Because of that, we have this complex over here, the PKA GLI complex, and also has SUFU here. These are kind of mediators. SUFU, COS2, and KIF7 are just kind of, they don't really have any, they're just to bind to the microtubules. They're like mediators or chaperone proteins, or like they help to form the complex, but they're not really doing much. Anyway, so we have this microtubule bound complex, and in this complex, what happens is GLI is phosphorylated by PKA, and that causes it to be dimerized or cut in half. When that happens, those half proteins are moved into the nucleus, and they act as transcription repressors. So in this natural state, when no Sonic Hedgehog is present, this microtubule-based complex is creating repressors, creating GLI as a repressor. And so when we look at Drosophila, it's the same thing. The proteins are different. So CI is the repressor. We still have PKA slimes in there instead. And some of these are slightly different. COS2 and KIF7 are the same. SUFU is in there. There's another one called fused. So they're very similar, but we're going to, like, for purposes of like the exam and stuff, we're going to focus on the vertebrate one. So now what happens if Sonic Hedgehog is present? So if Sonic Hedgehog's present, it will bind those two membrane catching proteins, which were the GAS1 Bach CDO complex, right? So it's going to bind those. What's going to happen is Hedgehog will then be brought into the cell with patched. It will be brought in with patched. And when that happens, patched can't stop smoothened anymore. So smoothened then will get phosphorylated, and it will come up and it will interact with this microtubule network and stop it. So since this microtubule network, remember, if it was not repressed by Sonic Hedgehog through that cascade, if this binding doesn't happen and then smoothened gets blocked by patched, then this cuts GLI in half and it acts as a repressor. But now, since that Hedgehog's here and it blocks patched, smoothened, gets phosphorylated. It blocks this COS2 KIF lysis apparatus or thing that was cutting gli in half, and it allows gli to still be imported into the nucleus, but this time as a whole protein. And when it goes into the nucleus as a whole protein, it acts as an activator of transcription. So instead of a repressor, like we saw in the last slide, it now activates transcription of genes. And so this then allows whatever developmentally important genes are under that promoter to be transcribed and then cause a different cascade. So when Hedgehog doesn't bind, gli gets cleaved in half and it acts as a repressor. When Hedgehog does bind through patched and smoothened, and this microtubule complex, it remains intact and it acts as an activator. So that's a quick Sonic Hedgehog example, and I'll catch you in the next one.